Continuing on, there is another technique called as radial or circular chromatography. Okay, so the plane of paper is kept horizontal. It is kept horizontal, but the solvent here moves laterally. Okay, towards the periphery, from the center towards the periphery. Okay, if you see the diagram here, radial paper chromatography, you can see there is a solvent fund, there is a circular filter paper which acts as the base. Sample is spotted at the center, okay, at the middle, instead of in one direction. If you see in paper, ascending and descending paper chromatography, sample spotting is done at one end. Okay. Unlike that, in radial, it is done at the center of the circular paper. And below that, the wick connects to the solvent system. Okay. So there is a cotton wick that connects the paper to the solvent system. So what happens here is very simple. The direction of the solvent flow, you can see it moves from bottom towards the paper initially to capillary action, and then from there it will move in circular fashion uniformly. Okay, so the movement of solvent in the chromatographic paper is uniform and circular. This is achieved by the capillary action. When we drop a liquid, a drop of liquid is added onto the filter paper, we see it uh, distribute itself evenly throughout the paper. So that is the idea behind radial chromatography. Other things remain same, okay, because of the coefficient of separation. The constituents of the sample get separated. Okay. So next part is spraying. So once what do we see here? Sometimes a non-chromogenic or non-coloring component, if it is present in the sample, once it is separated, there is no way of identifying the separation. Okay, because it is colorless, so it is not easy to visualize the separation unless some sort of coloring is added to. Okay, so there are this process of coloring, we can say it is spraying in this context. So when do we spray? After the movement of solvent to expected distance. Okay, whatever expected distance it is there, it is visible. In this case, it is 15 to 18 centimeter. Once the solvent has moved, we spray the paper. Okay. So when do we spray it? We remove the paper from the instrumentation. Okay. We make sure there is no more movement of solvent happening. We make sure that there is no connection of spraying to other chemicals that are placed. Okay, once it is removed, we have to make sure to dry the paper. If the paper is not dry, during spraying, there's a possibility that it can run or spread even more than expected. Okay, so paper is removed first from the instrumentation. Make sure it is dry. There are two ways of drying it. One, we can place it in an oven, which will uniformly dry it. But sometimes there's also an emergency or method to do when there is urgency for the result. We can use a hair dryer. But we prefer to do it oven because of uniform drying. Okay. I have to make sure that the drying should not be prolonged, it should not be too long. If it is too long, it will form a like crisp on top. Okay. It will form a layer which while spraying can dislodge from the paper itself. So you have to make sure that the paper is not too dry as well. Once it is dry, you have to spray it with the suitable reagent. Okay. So this suitable reagent can be any form of dye which is compatible with the product, so that it can easily visualize the distribution of analytes across the paper. That is the aim for us. We have to see what are the different types of uh, components that has been separated from the sample mixture. And selection of the dye is done so that it is complementary, chemically complementary to the component, so that it is easier for the distribution and easier for the identification of the molecule. It should not be in such a way that once you spray, it should not have a chemical reaction other than just a color reaction with the element. The next is very important. Rate of flow or RF value. What is that? So RF value, the definition goes something like that. It's the ratio of distance traveled by the solute to the distance traveled by the solvent front. What is solvent front? That is the movement of the mobile phase. The edge of the mobile phase that keeps on moving. The distance traveled by the solute will be from the point of application when we visualize the separation. So if you see here, let's say from here you have added, this will be the solvent front, okay, where the mobile phase is moving, and this is the distance of the analyte that has traveled. Okay, so it's a ratio between the movement of the solvent front and the distance of separated analyte. Okay, that we get distance moved by the solute divided by distance moved by the solvent front. Either we can use any units here, we can use centimeters or inches. We prefer to use centimeters because it's easier. So what does it characterize? What does the RF value indicate? It characterizes the distance moved by the solute in the direction of the solvent flow. Okay. As the mobile phase is moving, we have already seen the analyte will also get separated and move in the same direction. Okay. So RF value gives us a unity, gives us a number to check the distance that has moved along with the solvent. So how the solute moves along with the solvent, we can give a graphical or a numerical representation to it. Under standard condition, it closely reflects the distribution coefficient and it is characteristic of that compound. Now, this is where RF value becomes very, very important. Okay. Under standard conditions means when everything is controlled, there is no changes in condition happening during the process. We can say that RF value is almost similar to the distribution coefficient for that solute, and we know that distribution coefficient is constant and it is characteristic to that solute. It does not change. It remains same. So based on this, we identify what type of solute it is. So where do we use it? What is the clinical application of this method? So we commonly use it for separation of amino acids, peptides, alkaloids, different types of sugars, different types of lipids, etc. in the biological sample. So whenever we require to isolate a particular type of molecule from a mixture of sample, we can use chromatography, paper chromatography technique, because it is easier to perform and it is cheaper. Okay. So it can be also be used to a limited extent as a method for preparation. Okay, let's say you have one amino acid, you have to prepare one amino acid X solution. Okay, but this amino acid is only present in the form of mixture. So by this, what you can do, you can use the separation technique and to certain extent use this technique for preparation of solution of that amino acid. Next, to study the structure and amino acid composition of the protein, very important. Okay, just look at any molecule at the molecular level. See the structure. So see the basic composition of these molecules. More into research side. We can also use it for analysis of blood, hemoglobin, urine, which is of great diagnostic value. So mainly when there are disorders where one particular type of amino acid might be expected in blood samples or a type of carbohydrate may be expected in blood samples, it becomes very easy to isolate them and hence adds value to the diagnostic application of chromatography. Okay. Okay. Thin layer chromatography is the next part. So here, unlike paper chromatography, we have linear chromatography where it is performed on a sheet of glass or a sheet of plastic or an aluminum foil. So what do we see here? The support base is different compared to paper chromatography. In paper chromatography, the base, the support was made up of paper. On top of that paper, some other molecules were sprayed. Here, the support is made up of sheet of glass, plastic, or aluminum foil. On top of this support base, the stationary base needs to be constructed. Okay, so that is coated with a thin layer of adsorbent material. Okay, we should call it a stationary base. We can use silica gel, aluminum oxide or aluminum, or cellulose as our stationary base when we are using thin layer chromatography. Okay. Let me repeat what is thin layer chromatography. It's a technique which is performed on a sheet of glass, plastic or aluminum foil, which is used as a base for our stationary base. The stationary base is then coated on top of this base as a thin layer made up of absorbent materials such as silica gel, aluminum oxide, or cellulose. Here, what do we see in routine diagnostics? Okay, all over the district, wherever you see a TLC being performed, cellulose is commonly used as stationary base because of its easy availability and cost effectiveness. It is commonly used, but there are certain for certain analytes, particular type of molecules needs to be used, particular type of stationary phase needs to be used. So it depends from analyte to analyte. Okay. So in 1989, AI Vogel, he said following sample application mobile phase is drawn up to the plate via capillary action. So this person, AI Vogel, in 1989 gave us the principle behind how now thin layer chromatography works. Okay. So by this we can see it is quite a recent methodology. It's not very old. It's quite recent. Okay. So he says once sample is added, mobile phase is drawn up to the plate by capillary action, same like paper chromatography. Because different analytes ascend the PLC plate at different rates, separation is achieved. Okay. But here at this point we can see the principle of thin layer chromatography and paper chromatography changes. In paper chromatography, that separation depends on the coefficient of separation. Okay, distribution coefficient. But however, in linear chromatography, the separation happens based on the ability of any analyte to ascend. There are many factors responsible here which affects that ability to ascend. If the molecule is structurally bigger or has a more molecular weight, it will be obviously will be slower to ascend compared to one with lower molecular weight. Many things comes into play, but that is the basic principle difference in principle. One moves along with the mobile phase because of distribution coefficient. In here, like essence. Okay. So the proper principle. Thin layer is formed on a glass or plastic surface, as we have seen. The base for thin layer chromatography is either a thin sheet of glass, plastic, or aluminum foil. On top of that, the thin layer is constructed that becomes our stationary phase. The movement of mobile phase occurs generally by capillary action, and again it is capillary action because it is liquid. Here we can say most of the time it moves from capillary action, but when gas and liquid are involved as stationary phase, there is no longer capillary action, but it might require a pump. You don't have to worry about that because it's not included for you. So just right now focus that movement of mobile phase occurs generally by capillary action. As mobile phase moves from one edge to another edge, okay, as it is moving, the analytes are placed on the layer at a rate determined by the distribution coefficient. So based on the distribution coefficient, they are separated as mobile phase moves from one edge to the another edge of the stationary phase. So here, the movement of analyte is expressed by retardation factor, which is calculator. Okay. So what did we see in the last lesson? We saw RF value. Okay, rate of flow as our value to identify any analyte in paper chromatography. However, here in the chromatography, we use something called as retardation factor, which needs to be calculated. Okay. So, what are base or plates now? So, keep the plates clean. Okay. Make sure it is kept clean because once you apply the gel, it's not as easy as removing the paper. Once you apply the gel, it needs to be completely removed for many reasons. One among them is the chemical action or action between the sample and the stationary phase to get it. So make sure the plates are kept clean using chemicals and make sure these chemicals which are used to remove are not allowed to form any deposits. Okay. Once you have used the chemical to remove the stationary phase from the previous experiment, make sure that you also remove these chemicals which were used. No deposits are left behind after that. Dry it off completely. Make sure it dries completely. There is no residues remain. After that, don't avoid handling of the plate. Now, what do you mean by handling of the plates? Make sure that you don't press on the surface. Okay. Once you do it, the possibility of transferring whatever chemical components that are present from your finger onto the plates is very high. That is one thing which affects the analysis. Second thing is you might have to redo all the cleaning and drying again, which will waste lots of time. So this ensures that there is no fingerprint, dust or lint that are deposited or remains on the plate. So it's very important once it is dry, you make sure you are completely wrapping it. Okay, wrapping it with lint-free papers to ensure there is no deposits that are being formed. Now let us go into the stationary phase. You can use silica gel, alumina, cellulose, or gypsum. Gypsum is rarely used. Cellulose is commonly used as the stationary phase in a diagnostic laboratory. Often cannot be interchanged without modification of solvent system. This is very important. Each stationary phase has a compatible solvent phase, the mobile phase. Okay, it's very difficult to interchange the stationary phase with the mobile phase. Okay, the stationary phase, which is the mobile phase, which is prepared for silica gel, cannot be used for alumina or cellulose. Similarly, just you found out that there is no cellulose side in alumina, then you have to change the mobile phase for that as well. So these are linked. Okay, they are compatible, and it is very difficult to interchange. You can say it is impossible to use the same mobile phase for different stationary phases. So let's go into the technique. First, prepare the slurry of the stationary phase in proportion of the particular adsorbent that is being used. So we can get all this information from the protocol. We don't have to worry about this, but make sure the slurry is prepared for stationary phase in proportion to the particular adsorbent being used. Place the plates on level surface. These clean dry plates are kept on level surface without any vibration or any slant. So why this is important? If there is a slant, when you pour the slurry onto the plate, it causes uneven deposits. Okay, it will not uniformly set. One edge might be having higher concentration of the slurry. Another edge will be very thin. Somewhere it will be very thick. To ensure this does not happen, you have to make sure that the surface that you are going to place your plates is level. Once that is done, add the slurry on the plate. Allow to dry for at least 30 minutes. Okay, to ensure that the slurry is dry. Otherwise, what happens is there's a possibility that the areas of the slurry over the plate may be dry for a different extent. Somewhere it might be completely dried, somewhere it is half dried, somewhere else it might be uh, completely running. And please don't use any artificial methods for drying. Okay, don't use a hair dryer or don't keep it on a hot plate. Let it dry naturally. Once it is dried, sample is spotted onto the stationary phase and allowed to air dry again to ensure proper drying. In a tank, next in a tank, fill solvent system, that is mobile phase. Then lower the plate vertically. Okay. Make sure that only 0.5 millimeter is immersed into the solvent system. So this is a diagrammatic representation of how it is done. So once sample is spotted and dry, you are going to lower it into a chamber. The chamber contains the solvent system. Again, make sure the sample spotted is higher than the level of the solvent. It's not immersed. Okay. Then you are going to close it with a wash glass to ensure there is no evaporation. Okay. That is the test for 2 to 3 hours for movement of mobile phase in the capillary action. It takes longer. Okay. Here the time takes longer. It's very important that you give adequate time. 2 to 3 hours is preferable for movement of mobile phase. So once it is done, 2 to 3 hours is done, remove it, let it dry under warm air for few minutes. Okay, where do we get this warm air? Either from an oven or a hair dryer. Better oven because of uniform drying. Next is dyeing the plate. Okay, you have to dye to observe the separation. Adsorbent is inorganic, so we have to use a stronger solvent. Okay, concentrated H2SO4 is sprayed and followed by heating of the surface. So what is the advantages of PLC? It is versatile, faster, and more reproducible than other techniques. Okay, we can use it to separate different types of mixtures. It is relatively fast and it is more reproducible. You can get the same results again and again. Comparatively less expensive than other high-end chromatography technique. Separation can be carried out in large numbers. You don't have to worry because of this less expensive. More adsorbents can be used compared to paper. Being a strong base here and a inert base, it's very easy that we can use more and more quantities of adsorbent on top. So sample requirement is very minimal. It allows for adequate separation in small distance compared to paper. Okay, paper requires longer distance. This requires smaller distance. So clinically, it is used for screening procedures to identify certain abnormal constituents inborn errors of metabolism to identify different amino acids in the urine sample. Lipids can also be separated. Amino acids, bile salts, carbohydrates, drugs, porphyrins, prostaglandins, hormones, derivative of nucleic acids, etc. Other components can be separated. So why it is versatile? Because almost all the components, all the molecules can be separated using this technique. So these are the references from where I have taken the details.